Importance of leaf culture. Culture of excised leaf primordia is valuable to study the effect of various nutrient, growth factor, and changing environmental conditions on leaf development under conditions divorced from the complexities of the intact plant. In case of fern, leaf primordia culture are used to study the formation of sporangia and the size at which a primordium is destined to become a leaf. Young leaves of most of the Solanaceae species form numerous shoot buds instead of callus formation when they are cultured in solidified MS medium. When shoots have grown to a height 3 cm, they may be removed and subcultured onto MS medium devoid of growth hormones. Root formation is stimulated by this treatment. Therefore, leaf culture of Solanaceae species can be used as clonal micropropagation. Importance of shoot tip culture. The first point is virus eradication. Many important plants contain systemic viruses which substantially reduce their potential yield and quality. It is therefore important to produce virus-free stock which can be multiplied. Generally, highly meristematic tissue of a virus-infected plant remain free from virus due to their first mitotic activity. Therefore, shoot tip and meristem of a virus-infected plant are the ideal explant to produce a virus-free stock. This technique is also valuable for the maintenance of carefully defined stock of specific varieties and cultivar in disease-free state. The size of the meristem explant is critical for virus eradication. This technique combined with heat treatment or chemical treatment has proved to be very effective in virus eradication. Heat treatment is done by placing an actively growing plant in a thermotherapy chamber. Over a period of 2 weeks, the temperature is increased to 38 degrees centigrade inside the chamber and the plants are maintained at this temperature for 2 months. After that period, the apical meristem is excised, surface sterilized and transferred aseptically to agar medium. Using this technique, 85% to 90% virus-free stock have been obtained. Without heat treatment, shoot tip or meristem can be grown on chemotherapant added medium for virus eradication. Next is micropropagation. A sexual or vegetative propagation of whole plant using tissue culture technique is referred to as micropropagation. Shoot tip or meristem culture of many plant species can successfully be used for micropropagation. Next point is storage of genetic resources. Many plants produce seeds that are highly heterozygous in nature or that is recalcitrant. Such seeds are not accepted for storing genetic resources, so the meristem from such plant can be stored in vitro. Next point is shoot tip or meristem culture and plant breeding. In many plant breeding experiments, the hybrid plant produce abortive seed or non-viable seed. As a result, it makes a barrier to crossability in plant where non-viable seeds are unable to develop into mature hybrid plant. Shoot tip or meristem from such hybrid plant can be cultured to speed up breeding program. Next point is propagation of haploid plants. Haploid plants derived from anther or pollen culture always remain sterile unless and until they are made homozygous diploid. Meristem or shoot tip culture of haploid plant can be used for their propagation, from which detailed genetic analysis can be done on the basis of morphological character and biochemical assay. Next importance of shoot tip culture is meristem or shoot tip culture, and quarantine. There are some strict regulations concerning the international movement of vegetative plant material. After thoroughly checking, the plant material may be rejected by quarantine authority if the plant material carries some diseases. Plantlets derived from shoot tip or meristem culture are easily accepted by the quarantine authority for international exchange without any checking. Therefore, using this technique, crop plant can be easily exchanged in crop improvement programs that are based on materials from different parts of the world.